Hello everyone, this is Mrs. Hunt and this is Module 9. We are getting ready to start a new unit, so I'm really excited. The next 3 weeks we're going to be going over DNA. So let's go into some notes today. This is just a video over notes. So we're in Chapter 12. So I encourage you to read through Chapter 12's textbook. Make sure you're looking over your vocabulary. We're going to go into the notes of Chapter 12. And for Module 9, I'm only going to go over Section 1. So we're just going to do Section 1 this week. Okay, now for my face-to-face kids, I've got printed out notes. But if you're online or if you're absent, I would recommend getting some paper out. I have a copy of the notes for you in the Google Classroom. You can definitely use a PDF reader and annotate as you go through. If you have the ability to print it out at home, you can do that. Or you can just write your notes on paper as you go through here. Any kind of key things that we're talking about while the notes are going through. Okay, so again, this is a big chapter, but this week we're going over the DNA, the genetic material. We want to know what it is, what the shape is, what it's made out of, and go into the details of DNA. Next week, we're going to look at replication, and we're going to look at DNA, RNA, and protein. And then we're going to look at some mutations. So week 3 will be mutations. Okay, the beginning of our chapter is going to talk about several different scientists. These scientists are nice to know. Being an honors class, it's good to know the people who contributed towards the theories that we have and the evidence to support what we say in science. But you probably will not see these guys referenced on an EOC, an end of course exam. So while these guys are nice to know for worksheets and for information and to be able to talk about in class, it's not for the big knowledge for this week. Okay, so our first scientist is Griffith. Griffith is known for killing a lot of mice. And what he did is he injected the mice with basically a bacteria. And when the mouse got the genetic information, it died, and he heated it up. So what he was basically doing was creating a vaccine. He didn't know what he was doing, but what he showed was there was something being transmitted that changed, that was being passed from one thing to the next. So, and of course it ended up with the mouse dying. Right here though, so here he puts in the bacteria that causes a death to the mouse. There is another strand of this bacteria that doesn't kill the mouse, okay? The S strand that killed the mouse gets heated, denatured. We've talked a lot about that last week, and the mouse lives. But here, he puts both strands together. So he takes the denatured one that definitely killed, but he mixes it with the R strand. And what we find is that the R strand picks up the genetic information from the S strand. And when it picked up the genetic information, it ended up transmitting and killing the mouse. So again, Griffith wasn't sure what he had discovered. And this is 1928, by the way. He wasn't sure exactly what he discovered, but he knew it was something that was being transmitted. And they didn't have a name for it yet. But it led to the discovery of DNA. But he said that it was some kind of nucleic acid because we were able to identify it as that. So let's go back to our biochemistry real quick. Remember, we had carbohydrates, lipids, protein, and nucleic acid. So nucleic acids had carbon, hydrogen, oxygen, nitrogen, and phosphorus was in there. So that we could identify and we could label it a nucleic acid. Now, what kind it was, what was it doing, they weren't sure about that. We know that at this time that, children look like parents, that, traits run in families. We understand that, but we don't know about what the genetic information is. So they haven't determined exactly if it's a protein, nucleic acid. So this was the first where we've got it's a nucleic acid. We don't know what it looks like. We don't know the shape of it, but we're seeing the transmission of a nucleic acid. Okay, the next scientist is Avery. And Avery came through and he identified the molecule that was transformed in the R strand and the S strand. And he concluded that when the S cells were killed, DNA was released. So by denaturing and killing off that strand, the DNA was being released, and that was being picked up by the R strand. So again, it was genetic material that was being transmitted. Okay, our next scientist is Hershey and Chase. This is Alfred Hershey and Martha Chase. This is 1952. So I think this is really significant that we have a female, Martha Chase, that's being listed on this experiment in the 1950s. That was not very common for females, especially to be in chemistry, but also to be given credit for this discovery. Hershey put his name with Chase, so it's the Hershey Chase. And like I said, that's a female, so that's pretty cool to think about a female leading this. These two used radioactive labeling to trace DNA and protein. And they concluded that the viral DNA, so they did this in a virus right here, and they used E. coli bacteria and a virus. And they concluded that the viral DNA was injected into the cells and provided the genetic information needed to produce the new virus. So they injected the viral DNA, we didn't know it was DNA, genetic material, into the E. coli, and it was transmitted and passed down further. And they used radioactive labeling so that they could identify that this was the trait that was following through. What they did, they supported everybody else's stuff or information that this was a genetic information that was being passed down. They supported that it was a nucleic acid that was being passed down. So they helped back all of the previous research up. Okay. All right. And this is going to lead us to our DNA structure. Let's talk real quick about the structure of DNA. We're going to know that DNA is made out of nucleotides. So remember nucleic acid, the building block, the monomer was nucleotide. You have to know nucleotides are made out of three things. They're made out of a sugar, a phosphate, and a nitrogen base, a nitrogenous base. So you need to know that the nucleotide that builds nucleic acids consists of a 5-carbon sugar, here's the sugar, consists of a phosphate group, and consists of a nitrogenous base, the nitrogen group. So 3 things: phosphate, sugar, nitrogenous base. Okay. All right. So the next scientist, Chargaff, I don't have him written down. I don't know what the date was. Chargaff is going to be the next scientist. So we've got Hershey and Chase saying it's genetic information. They help us even contribute towards that nucleotide, 3 parts to a nucleotide. Chargaff is our next scientist, and he starts looking at those nitrogen bases, those nitrogenous bases. And he discovers that there's really only 4 nitrogen bases. In middle school, you guys learn these as A, T, C, and G. Okay. You still have to know them in order, A, T, C, and G. Just know that A and T go together and C and G go together. Okay. Chargaff's rule was that the A and T are in similar quantities and the C and G are in similar quantities. Okay. So that means that they must match each other. So Chargaff's rule is that C binds with G and T binds with A in equal parts. So these two go together and these two go together. So Chargaff's rule is going to pair up A and T and C and G. I remember DNA is where it's at. A binds with T. And then, of course, C binds with G. That makes it a little easier. Okay. All right. So up until this point, we know it's genetic material. We know it's a nucleotide. We have we know it's nucleic acid. We have what a nucleotide is. We have Chargaff's rule with the base pairs for the nitrogens, the nitrogenous bases. So we're getting closer to discovering DNA. All right, this is Rosalind Franklin. Rosalind Franklin took this picture in 1951. Let me move my picture here. So Rosalind Franklin, first of all, she's a female. That's really big. She was studying, she's a physicist, and she's studying DNA, genetic information. And she takes this picture using an X-ray diffraction machine. It puts out a lot of radiation. Unfortunately, she takes so many X-rays that she ends up giving herself cancer. She didn't know that. They didn't know that cancer was caused by radiation. So she ended up dying at the age of 37, which is really terrible. But she took this picture. She went to several science conventions, and she said DNA is in a double helix, that twisted ladder, that spirally shape to it. And this is really significant because up until now, we don't know what the shape of the molecule is. We're getting the components of it, but we don't know it's that twisted ladder. We don't know how it replicates, lots of questions. But she says it's a twisted ladder, and she matches Chargaff's base pair rules. She matches what a nucleotide is and the parts of the nucleotide, and she comes up with her model of DNA. Okay, unfortunately, she dies after presenting her data. She presents it to these two fellows. This is Watson and Crick. Watson and Crick are given credit for creating the model of DNA. Here you see their big giant model of DNA. And that gives us the twisted ladder right here. And it matches everybody else's previous discovery. It is a nucleic acid. It is made out of the nucleotides. It has Chargaff's rule, A binds with T, C binds with G. It is made of a sugar phosphate backbone. So they've come up with the model of it. Watson and Crick build the model. They present it. They win the Nobel Prize. Rosalind Franklin had already passed away at this point, and you can't win a Nobel Prize if you have passed away. So she did not win the Nobel Prize. Watson and Crick didn't really give her much credit. They said there was this lady who took a picture, and that's about it. So it's kind of sketchy in history that these two guys kind of got all the credit, but they did. So in our textbooks and everywhere, it still says that Watson and Crick model of DNA. I do appreciate that Rosalind Franklin was mentioned in the book, and that she's the one who took the picture and basically came up with the construction of it. All right, so Watson and Crick are going to build their model. They have two outside strands consisting of alternating deoxyribose. That's why we call it DNA, a deoxyribose. Now, you should know deoxyribose, ose, is a sugar. So that's your sugar. Deoxyribose is the sugar in DNA. And the phosphate group, that's the P in the phosphate group. Okay? And so you can tell people that you are made out of sugar. All of the DNA inside of you is made out of sugar. So you're sweet as sugar. All right, in the middle, the ladders, the rungs of the ladder is cytosine that pairs with guanine. Those are the base pairs that bind with each other. And they have 3 hydrogen bonds in between them. So the C and G bind together, and those are hydrogen bonds. Thymine binds with adenine, and this forms the base pairs, and they have 2 hydrogen bonds between that. So C binds with G with 3 hydrogen bonds. Okay, I'm going to try to write this down real quick for you. And T binds with A with 2 hydrogen bonds. So there we go. So we write it like this. We put the 3 hydrogen bonds between the C and the G. And then adenine and thymine, we put the 2 lines between there. So that's representing those hydrogen bonds. So 2 and then 3. Okay. DNA is often referred to as a twisted ladder. The rails of the ladder are represented by the alternating deoxyribose and phosphate, so that's the sides of the ladder, and the rungs are in between. And these are the base pairs. Cytosine pairs with guanine, and thymine pairs with adenine to form the steps. So this is kind of on the side here. And of course, it gets more complicated because it is. So here's the adenine and thymine, and you can see that they've paired with 2 hydrogen bonds. You see the C and the G, the cytosine and guanine, and they've paired with 3 hydrogen bonds. All right. And then you see on the side, we have a phosphate bind to a sugar, bind to a phosphate, bound to a sugar, phosphate, sugar, phosphate, sugar, phosphate. Now, if you kind of notice that this one up here, this is a 3 prime, and it goes to 5 prime, and the 5 prime to 3 prime. So let me explain this real quick. If you look at one side, the 3 to 5, it's going sugar, phosphate, sugar, phosphate, sugar, phosphate, and it's running in a particular direction. The 5 to 3 is actually upside down. It flips it. So I'll show you right here. This is a little bit better of a picture here. Okay, so here we go to the 5 prime to the 3 prime end, phosphate, sugar, phosphate, sugar. And then the other end is a 3 to 5 prime, and you can see that they're upside down. Okay, so the strand that runs, here we go. On the top rail, the strand is said to be oriented 5 to 3 prime, and the strand on the bottom is running the 3 to 5 prime in the opposite direction. The strand of DNA has 2 ends, okay, and DNA is always copied from the 5 going to the 3. So we end up calling this side the leading strand, and the other side we call the lagging strand. Okay, let me see if I don't have that up there. Okay. So when they go to copy, which will be next week, we'll talk about replication. They're going to be in a different pattern. So all we want to work on this week is building our model of DNA, making sure that we've got the base pairs, that we have the phosphates and sugars, and then we know what a nucleotide is, the phosphate, sugar, and nitrogenous base. Now, I'll go ahead and give you some extra things. They are going to, when your body reads a code, it reads these 3 letters together as a code. And that code codes for an amino acid. Amino acids get put together to build a protein. So, for example, I have brown hair. The color of my hair is on my genes, it's on my DNA. The length of my hair is not on my DNA. That's just my personal preference. I like to have longer hair. But the brown is on my DNA. So my DNA would code 3 letters to build build an amino acid. More letters code to build another, more, to build a protein that codes for brown hair. All that is on my DNA. So my children will inherit my DNA, and they're going to get what's passed on from my generation or from my DNA onto my offspring. Okay? The DNA itself, I just want you guys to understand, the DNA itself is twisted up. It's twisted into these things called nucleosomes that go into histones that go into nucleosomes. Okay, my DNA is wound up around a protein. DNA is so big. There's so much information that the only way for it to fit in the nucleus of a cell is it needs to be compact. It needs to be squished. So we're going to twist it up, and then we're going to wind that around a protein, a histone. And then that protein is going to be wound up again around a nucleosome. And that nucleosome is going to be super coiled up even tighter. You can see it right here. We're going to turn it into a chromosome. So this is a chromosome right here. Together, this forms a chromatid. And we're going to get into all of this when we get into more of the genetics and meiosis and mitosis. But that DNA is our genetic code that codes for you, that codes for your height, your skin color, your eye color. We're going to be talking about some things that you inherit from your DNA. So that's pretty cool. So if you take your hands like this and clasp them together, don't even think about it, just do it. Which finger's on top? Mine's my right on top. That's actually on my DNA, which is weird. Finger clasping is on my DNA. If you do it the different way, if you try to move your fingers down to the other side, it feels really weird. It's like, this is not right. This finger's dangling down. It just doesn't feel right. But the interlocking of my fingers is actually on my DNA. All sorts of cool stuff is on our DNA. And then, of course, there's other things that we're going to learn from our environment. The reason why I have a Southern accent is because I'm from Tennessee. All those kind of things are our environment. The length of my hair is just because of a personal preference. So we're going to figure out what's on our DNA and then what is our nurture around us. It's playing a role. All right. I think this website is super cool. I am a child of the 90s, was born in the 80s, and they were working on the Human Genome Project for years in the 80s. And this is the poster for the Human Genome Project. And we can scroll through here and dive into it and zoom into it as best you can. So spend some time looking through here. These are all of the codes on the human genome that code for a trait. You can find things like red hair. You can find inherited diseases. Oh my goodness, there's a lot of diseases. So you can definitely see all the different chromosomes that you would inherit. You get 22, and then your last 23rd chromosome is your gender. Girls, you are XX, and boys, you are XY. And we're going to get into all of that even more. But this is really cool to spend some time. We'll look at it in class. I'll pull this up and let kids zoom into it and discover some things that are inherited on your DNA. But this is in every cell of you. It's in every nucleus and of every cell. And this is also in our reproductive cells, the egg for females and the sperm for males, that's going to be passed on to our future generations. You inherited your DNA from your parents, and you got a copy from each. So it's really cool to think about all this. This was a huge science project that was put on around the world to discover all the traits. Look at this one. Chromosome number 3 has 199 million base pairs. That means 199 million. A binds with T. That's 1. T binds with A. That's 2. C binds with G. That's 3. G binds with C. You kind of get how this works. 199 million base pairs. And then they had to figure out, what is that code for? What is that? What is it saying on our DNA? So some pretty cool stuff. Okay. And the last little bit I will leave you is not everything is on our DNA. Some things like Down syndrome will be on the DNA. You can definitely inherit Down syndrome and you can pass it on. There's also environmental factors. So Down syndrome is also contributed as a mother ages. Her risk of Down syndrome also increases. So there's other things that contribute towards genetic illnesses besides just passing it down from your DNA. All right. And then so next week, we'll get into section 2. So this week, read section 1, chapter 12, go through these notes, and we're going to work on building that model of DNA this week. And I hope you have a great week.